Right, so this is monoclonal antibodies topic, which is in the year 11 triple biology. Okay, so it's a little bit of a topic. It kind of links to the disease and defense topic, but we're going to just do the little bit that is extra for you, which is monoclonal antibodies. So to get you started, like we have been doing all along now, on your workbooks, in your booklets, on your piece of paper, however you may be doing it, I want you to write the date, whatever your date may be. Monoclonal antibodies, which is the title. There are 5 keywords, so you need to write them up, and then there are also 5 questions that you need to answer for your do now activity. So you need to press pause and have a go at the questions for your do now activity. Okay, they link back to the circulation topic, so the heart from core biology, so you should have done them before. Obviously, if you aren't sure, you can have a quick Google, go on BBC Bite Size, but also just wait for me. I'll give you some time to have a go, and then I'll start putting up the answers. Right, so the 4 parts of the blood, if you can remember from last year, you would have red blood cells, white blood cells, plasma, spot that, made that look a bit weird, and platelets. Okay, so they're the 4 types of blood. I've just realised that you can't quite see this. Okay, what is the function of the red blood cell? The function of the red blood cell is to carry oxygen. Okay, that is its function. It is to carry oxygen. Name the 2 parts of white blood cells. Okay, so the 2 parts of white blood cells for anyone who was feeling a little bit clever are there. Okay, so it's phagocyte and lymphocyte. They're the 2 parts of the white blood cell. What is the function of a phagocyte? Okay, to engulf bacteria. Okay, so to engulf bacteria. You might see to ingest bacteria. Okay, it's not just bacteria, it's other foreign microorganisms, but bacteria is most probably the most common one that you will come across. Lymphocytes then. Lymphocytes are our immune response, so they're what makes our antibodies, our T cells, all of that. Okay, so our immune response, which is quite relevant at the moment with all certain diseases floating around. So, nod words, your learning objective, you might want to note this down just so you know what you're doing today. Evaluate the uses and formations of monoclonal antibodies. So we're first going to look at how what it is and how they're formed, and then we're going to look at some of the uses, because there's quite a wide range of uses that we would use them for. So, just as a little bit of a background, what are antibodies? So I'll zoom in on this a little bit. Antibodies are proteins produced by a type of white blood cell called lymphocytes. We just talked about that. You would have done a little bit of that in the heart topic in year 10. Pathogens have proteins on their surface called antigens. So if we're looking over here, antigens are the structural part of the virus. Okay. So all on the outside here, these are the antigens, okay? This is just kind of what coronavirus looks like, so it's a little bit relevant at the moment. So these on the outside are the antigens, okay? A virus would use them to bind to the cell wall of whatever cell it is, so in the human, and that is how it would infect, okay? So they use their antigens on the surface to bind to the cell wall of a cell to infect the cell. It might be the cell membrane. This must just be an example of a plant. Okay, so if it's in a human, they wouldn't have a cell wall, so it would just be the cell membrane. Okay, what an antibody does, okay, is it binds to them antigen spikes, so it stops it working. Okay, antibodies are specific, so an antibody is specific to a certain antigen, which means it's specific to a certain disease. Okay, so you have antibodies for every disease. Or you gain antibodies for every disease that might end up in your body. Okay? When a pathogen infects the body, the lymphocytes recognize this, the antigens as foreign, and attack them by producing antibodies. Okay, and that's what we looked at down the bottom here. Antibodies bind specific antigens, just talked about that. This means there's only one type of antibody in your body, okay? So that's why sometimes you struggle to get rid of certain diseases, because you don't have the antibody for it, okay? Also why vaccines are so important, because they help your body produce memory cells, which makes the antibody response faster, okay? Antibodies destroy the antigen, which is then engulfed and ingested by phagocytes normally, okay? White blood cells can also produce toxins that might kill destroy some of the antigens, okay? So just a real quick go back through, okay? Antigens are on the surface of the pathogen. So whatever's trying to infect your body, okay? They normally bind to something on the cell membrane. If you're a human, what the antibodies do then is they bind to that antigen instead, so it can't bind to the cell membrane, can't infect your cell, okay? Normally it's then transported to a phagocyte, which will then engulf it and get rid of it. Sometimes your white blood cells release toxins that can kill it, okay? So antibodies are part of your immune response. They fight infection. So as our first little review, okay, I want you to press pause and have a go at answering these questions. So write review at the top. And have a go answering these 3 questions. Right, so the first question for you was to describe an antibody. An antibody is part of your immune system. So it defends your body, normally attaches to pathogens. Okay, and it's the antigen part of a pathogen. Okay, it might be worth noting that they are specific. Okay, so if you've got an antibody for the cold, okay, or the flu, you've only got one antibody for that. Okay, if you then end up with getting measles or something, then you need another antibody. Okay, I'm just trying to think of diseases off the top of my head. Describe pathogen is something foreign. Something foreign that ends up in your body. Okay, something foreign that ends up in your body. Normally, bacteria or a microorganism. So something foreign that is inside your body. Its job is to invade. Okay, so it doesn't really want to hang around. Well, it does want to hang around. It wants to invade your body. Okay, describe how antibodies work when something foreign appears. So they attach to the surface antigens on the pathogen, okay, which stops them invading the cells. So it stops them invading the cells. Okay, so moving on now to something called monoclonal antibodies. Okay, so mono means one, and clone means an identical copy. Okay, so monoclonal antibodies are therefore identical copies of a certain type of antibody. Okay, and they're used throughout our system to produce certain antibodies. Okay, they're used quite a lot in cancer treatments because we don't have certain antibodies in our body readily available for cancer treatments, so they're used quite a lot in cancer treatments. There's an awful lot of ethics behind this as well, which I'll talk about in a minute, because we use the spleen of a mouse to produce these antibodies. So obviously they have a great cause in treating cancer and other diseases, which I'll talk about in a minute. It is kind of against animal rights because we're looking at using mice mainly to produce these antibodies that are then extracted for our use as humans. So the first thing that happens is an antigen, so that pathogen, that's something that's foreign, is injected into a mouse. Okay, so injected into a mouse, okay, it's vaccinated with a pathogen, and the idea behind that is it'll start an antibody response. Okay, so it'll start your white bloods working, you'll produce antibodies, that's the hope. Okay, it says there the mouse naturally produces lymphocytes which produce antibodies specific to the antigen. So they're producing antibodies that are specific to that pathogen, that antigen. Okay, spleen cells which produce the lymphocytes are then removed. So the mouse's spleen is then removed to get the antibodies out. Okay, so say we've injected this mouse with a certain type of cancer. This mouse then has started an immune response towards that cancer, produced antibodies against that cancer, and we are then extracting those antibodies from this mouse's spleen. Okay, then they are fused with a cancerous white blood cell, okay, because if you know much about cancer, they do not stop dividing. So the idea behind this then is that the cancer cells will divide and divide and divide, so you've got hundreds of antibodies because they've been injected with this cancer cell. Okay, it says white blood cells called melanoma cells form hybridoma cells which divide indefinitely. So you're making hundreds of antibodies because they're divided indefinitely. Okay, produce millions of monoclonal antibodies specific to the original antigen. Okay, so this is a quick way to make antibodies for a certain disease. Okay, but it's obviously using a mouse. It's also then fusing them with a tumour cell, so you can make loads of them because tumour cells reproduce over and over again. Okay. If you wanted to pause the screen and kind of look at it, okay, so you inject the mouse, it starts to form antibodies, the antibodies are extracted, kind of like fused with a tumour cell because tumour cells, so the antibodies are then fused with a tumour cell because tumour cells reproduce indefinitely. You produce hundreds of these antibodies and then you can use them for other things, okay, and we'll talk about their uses in a minute. So, your next little review then, just a bit of a fill in the blanks, okay, again, I want you to pause the screen, okay, and have a go at these questions. Okay, so an antigen, they've left it, yeah. So an antigen is injected into a mouse, okay? The mouse naturally produces lymphocytes, which are part of your white blood cells. So it'll naturally produce them. It's just starting an immune response, which produce antibodies specific to the antigen. Okay, spleen cells, which produce the lymphocytes are removed during a small operation. So this mouse undergoes a small operation to remove all its white blood cells, essentially, or its antibodies. Spleen cells are then fused with human cancer cells. These then form a hybridoma, so it's like a hybrid. Okay, the heart, the spelling's there. These hybridoma cells divide to produce millions of so a clone, monoclonal antibodies. Okay. So the next thing we're now going to look at is kind of like the true or falses. Okay, and I put a little bit at the bottom about like ethical issues as well. True or falses, advantages and disadvantages, what I mean. So advantages of monoclonal antibodies, even though they're expensive, they are cheaper than developing conventional drugs, so like antibiotics towards certain diseases, okay? Because it's based on tested technology, okay? So we're not having to do clinical trials because we know this works, because it's using essentially someone's immune response. Side effects can be treated and reduced by using mice, human, hybrid cells, or using a certain amount of the antibody. They are used in different ways, okay? They can bind to specific diseases, so they're very specific, and they can be used in a wide range of conditions, which we're going to talk about, where you're going to kind of do some research for me about it, okay? Disadvantage, they are quite expensive, and there are side effects, okay? Depends, you've got to outweigh how much the side effects affect you in terms of not having a certain disease. It's sometimes difficult to find the right antibodies. The mouse might, because we aren't mice and we have different kind of cells, it might produce a different antibody, or an antibody that can't be used within a human. So there is that little overlap there that we might not be able to find the right antibody. Okay, and I put down the bottom an ethical issue. Okay, ethics. An ethical issue is one that people disagree for either a religious or another moral reason. Okay, some people disagree with this some people disagree with the use of animals to produce antibodies. Okay, so that is a ethical issue. Okay, so review number 3, I'm just going to kind of leave you to it. It's a little memory test. I want you now, without going back, okay, have a think how many disadvantages and advantages you can remember, and then if you have to go back, you can then go back and write down the ones that you haven't remembered. Okay, so that's kind of like review number 3. I'm not going to put the answers up because you'll have the PowerPoint and you can kind of flip back. And then finally, what I'd also like you to do is a little bit of research. Okay, so there's quite a lot of uses of antibodies, monoclonal antibodies. I put up maybe the 5 most commonly used ones. Okay, so what I'd like you to do now is do a little bit of research, make me like a little mind map. So just tell me how they're used in pregnancy tests and just fill it in. Okay, how are they used in cancer treatment? Okay, so you've got 2 reviews kind of to do. I'll put the PowerPoint up for you. You can obviously fill it in your booklets or however you want to do it and then hand it in. Okay, thank you.